Today's lab is going to cover enzymes. Recall that enzymes are proteins, and these proteins are able to catalyze reaction. So first, let's look at the type of reactions that exist. There are two types of reaction that we're going to look at. The first one is endergonic reaction. In endergonic reaction, the chemical reaction absorbs energy from its surrounding. So in order for this reaction to occur, it requires energy in order for the reaction to move forward. So this reaction is not spontaneous because energy is required and must be absorbed. In this reaction, we start at a lower Gibbs energy for the reactant compared to the product. So for this reaction to happen, energy is needed in order for us to be able to overcome the amount of energy required to produce that product. An example of this reaction is photosynthesis, where in order for photosynthesis to take carbon dioxide and water and convert it into sugar, we require energy for this reaction. The energy comes from the sun. So the sun provides the energy in order to move the reactants, which is carbon dioxide and water, into the final product, which is going to be the sugar, the glucose. The other type of reaction we're going to look at is exergonic reaction. Exergonic reaction does not require energy. It actually releases energy as we move from reactant to product. So you can see here we start at a higher Gibbs free energy, and by the time that we get to our product, we have released energy, right? The product is at a lower Gibbs free energy compared to the reactant. This energy is spontaneous, and energy is released. So an example of this type of reaction would be in cellular respiration, where we take energy that we have stored and be able to break it down to release energy. Recall that in a reaction, so let's just draw a really quick sugar molecule right there, that there's a lot of energy in these bonds. So when we break these bonds, we are able to release a lot of energy out. So how does enzyme play a role in regards to reaction? Well, oftentimes a reaction, even if it's exergonic, doesn't always happen spontaneously because the different reactants need some assistant, and enzyme is this assistant. Enzymes can function as catalysts to help drive these biochemical reactions. One of the ways that they can do that is to help bind the different reactants, or also known as substrate, onto their binding site. This allows these substrates to find each other a little bit better and for the reactions to move forward. So here we have these two substrates, right? And they're just floating around, and eventually they'll find each other and their reaction can form so that they can produce this product right here. But oftentimes the conditions just aren't ideal, and yes, it will eventually happen, but it's going to happen very slowly. So what enzymes do is enzyme helps speed along this reaction. The enzyme will come along, and it basically is like tells these two substrates, hey, you guys should meet. So it grabs the two substrate into their active site. This area right here is active, right? And the active site is specific for the substrate. So it's going to grab this red substrate and place it here, and this yellow substrate and place it here. And so now you have these two substrates next to each other. The enzyme is helping move the reaction along faster because now the substrate can react and can work together in order to produce this product. After the product is produced, the enzyme, which is unaltered, will leave and then it'll go find another set of substrate. So enzymes are useful because they help push a reaction forward so that it can occur faster. So here we have that same chart that we looked at before. Recall that in the original picture that we saw that the Gibbs free energy was here, and this is an exergonic reaction, right? So the reactant starts at a higher level than product. So if the enzyme wasn't here, would the reaction still happen? Yes, it would probably still happen, but at a much slower rate. So by having the enzyme, we can actually lower this hump right here. This hump is also known as the activation energy. So without an enzyme, the activation energy is much higher. Once we add the enzyme to the mix, the activation energy is lower. This is the green line right here. It's lower. And so this reaction is more likely to happen because the enzyme helps orient the substrate in a favorable position. And by orienting the substrate in a favorable position, it helps for the reaction to occur. And so we have to undergo a smaller hill, that is less activation energy, in order to move this reaction forward. Now, remember that enzymes are protein. So proteins have a very specific 3D structure. Why is this important? Well, recall right here when I said that this active site is specific for particular substrate, it's because there's a specific structure, right? So you can kind of think of this as a lock and key situation, where there's a very specific key that kind of fits into this active site. So this 3D structure right here that we're looking at, this 3D structure has a very particular structure that's meant for this molecule right here to be able to actually recognize and bind into. So proteins have a 3D structure that can be influenced by different factors. And what do I mean by influence? Meaning that the enzyme has a particular pH and a particular temperature, a particular level of salinity that it likes to live in, right? So think of our body. Our body has a very specific temperature as well as pH. And the enzymes that are functioning in our body, those are their ideal pH and temperature. When you change the pH or the temperature, what do you think will happen? Well, in cases where you change the pH or the temperature, it can result in the enzyme not being too happy in that environment anymore, and it can actually cause the enzyme to change its structure, also known as denature. So it can cause the enzyme to denature, and it loses that same 3D structure. And once it loses those 3D structure, what else will happen? Well, it's going to have a hard time for the substrate to be able to interact with its active site, right? Because the structure has changed. So now it doesn't have the same formation. And so that active site is not going to be able to bind onto the substrate the same way it did before. It's lost its function. So you can think of like, now you have a key. Well, you can't just go to your neighbor and stick that same key in their door and expect it to work. Their structure is very different. So the enzyme, if you place it in a different pH, that can affect the enzyme structure, and therefore it won't function the same way anymore, and it's not going to be able to help substrate turn into product the same way it did when it was in its ideal temperature. Let's recall that enzymes have 4 different structures. The primary structure is just the amino acids themselves, right? Recall, amino acids are the monomers of proteins. When you combine multiple monomers together and you connect them by peptide bonds, they will form a polypeptide, which is the polymer of proteins. So here we have a polypeptide, which is just the amino acid linking. When we move to secondary structure, the amino acid now start interacting with each other. Amino acids have different R groups, which are just different molecules that are on the amino acid. And they can be as simple as a single hydrogen, or they can be more complex and actually be an aromatic ring or other molecules. So these molecules have very particular characteristics. And so now some of these characteristics of the different R groups can interact with each other to form either a beta sheet or an alpha helix. Once you have more complicated bonding between those amino acids, we move from secondary to tertiary structure. Tertiary structure is the folding of a single polypeptide. So now, in addition to the alpha helix and the beta sheet, we also can have other bonds that are happening. We have van der Waals bonds, we can have covalent bond, we can have disulfide bridge. So a lot more bonding is happening here in the tetrahedral structure. Proteins are rarely made of just a single polypeptide. So a functioning protein oftentimes is made of multiple polypeptides. So we can see here in this quaternary structure that you have two different polypeptides here interacting together. And they will be interacting also by different type of bonding, including hydrogen bonds, and function together for the protein to be a functional unit. So when you have this protein in its ideal environment, it has a particular shape and is able to interact with the substrate. Once you denature it, for example, by taking that protein and placing it in a different heat environment. So think of your eggs. Remember that egg white is made of protein, right? We always hear fitness guru tell us how we can eat egg whites for protein. Well, when you stick that egg white onto a frying pan, that heat changes that egg white from that more liquid state into the solid state. What's happening there is that protein is being denatured. So here, once we add, let's say, for example, temperature to this protein, it will cause the protein to change its characteristic. And so it will denature. And so that same structure that I had before, right here, is what we're looking at, that is no longer available on this protein because you have part of the substrate, the part that the substrate recognizes is here, and the other part that the substrate will recognize is over here. So they are no longer aligned in a way that allows that protein to interact with the substrate. If the protein can't interact with the substrate, well, basically nothing happens, right? Because the enzyme is no longer able to function as a catalyst to move that reaction forward. So in this lab, we're going to do two different experiments that kind of shows how enzymes functions. So the first one is going to be the renin lab, in which case the renin is the enzyme, and we'll talk more about that in the next set of videos. But the renin is the enzyme. We're going to be looking at temperature and how temperature can affect the enzymes. The second experiment is going to be about surface area and how the enzyme can function faster if it's able to actually find the substrate. So we'll look at how surface area affects reaction time. And the last experiment, which is not in your lab manual, but I'm going to demonstrate to you, is just looking at how can we detect pH. All right, I will see you in the next video.